Hi there and welcome. Today I'm going to talk about TRANSFAC BASIC package. Basically what it is good for, and what you can achieve when you have it. If you are working in genomics, in transcriptomics, or even in proteomics, or just analyzing your regulatory regions of your favorite genes, I think TRANSFAC BASIC package could be the choice for you for the everyday work. But let's have a look, and we'll start from overview of the features, like a big picture. It includes actually the TRANSFAC database, the world's biggest database on transcription factors and their binding sites, the golden standard. And it includes many different tools which you can use to actually do many things. First, you can analyze your data, your promoters to search for binding sites of transcription factors. Then you can actually search for enrichment of those sites and also combinations of sites in promoters of your genes. It also can be used for integrative analysis of various omics data. If you have RNA Seq, ChIP-seq data, or TAC-seq data, you can use it and you can analyze this data starting just from raw data or from pre-processed data. It has tools which apply different machine learning and pieces of artificial intelligence. Very easy graphical user interface for no coding bioinformatics. You basically don't need to code anything. You just drag and drop things and you run your analysis. You have a lot of tools, like more than 200 different tools and pipelines. Everything done in the cloud storage, and you got complementary 100 GBs for your data and result. Moreover, these tools have API which you can use to run those tools just from your computer using either Python or Java or R. Okay, now we go online. Here I have my username and password, and I'm subscribed to TRANSFAC BASIC now. So I see TRANSFAC here and the tools which we are going to check just in a moment. First, we go to the TRANSFAC database. So in the search interface, you can look for instance, for a given gene. For instance, take BRCA1. And here you can look at the information about this gene, how this gene is classified in gene ontology, and very important, we have the tissue expression of that gene in different tissues. And we have actually information about transcription factor binding sites experimentally proven in the promoter of that gene. Here you have you see the scheme of that promoter, and information about each individual site is collected from a particular paper, so you can see the experimental details about each particular transcription factor binding site, the location of that site in the promoter of that gene, basically which transcription factor was found to be bound to this binding site, in which experimental source and conditions, and also there is a link to the reference. Additionally, we have fragments which were detected by various ChIP-seq experiments in the regulatory regions of that gene, and this information is coming from ENCODE data or from particular publication. And also in each of the peak we actually detect the best match for a transcription factor motif which was taken from our database, from TRANSFAC database. As for the protein information, we have information about pathways where this protein is involved, collected from public sources like Wiki pathways or other databases. We have our own pathways curated in TransPath database, which this information you can access when you license TRANSFAC pathways. So for the transcription factor activity, because BRCA1 is also a transcription factor, you see the information which genes are regulated by BRCA1, and very important, we are collecting motifs for the transcription factor. So here we see two motifs collected from different sources. First motif was built using ChIP-seq data, from in vitro ChIP-seq data, and the second motif was built from ChIP-seq data. So when you click on that motif, you get more detailed information about the motif itself. So the motif in two orientations, also the source of that motif, basically how this motif was created, how this positional weight matrix was created, and the references which were used to build this matrix. The library of positional weight matrices is actually the foundation of TRANSFAC database. So the TRANSFAC BASIC includes over 10,000 positional weight matrices covering animals, plants, and fungi. And that's actually more than any other database, free or commercial. Why there are so many positional weight matrices? And especially you can see that one factor may have quite many motifs related to that factor. Because transcription factors behave differently depending on the cell type, the species of course, and the environment and conditions, other cofactors, this so-called redundancy actually reflects the real diversity of transcription factor binding site and transcription factor binding behavior. Basically, more positional weight matrices, better chances to find real functional sites in your data. So, for instance, when we look at the positional weight matrices collected in TRANSFAC for the P53 transcription factor, you can see so many, 12 entries, and these are, positional weight matrices collected from different papers, which were based on different types of experiments. Some of the matrices we built ourselves, some are taken from other databases. Next, I'm going to talk about MATCH, which is our core algorithm for site search and site enrichment analysis. MATCH uses a smart similarity scoring system based on something called the information content vector. This gives more weight to crucial positions in the motif, making the predictions more accurate. But MATCH goes even further. It can automatically adjust cutoffs to detect the site enrichment. It will save you from trial and error and save it from setting up some arbitrary cutoffs. I will demonstrate now how to run MATCH, and to do that you have to open geneXplain platform. Today we will take a simple example. Actually, it's a list of genes which were found to be upregulated upon treatment of cell line with TNF-alpha. And here we have 58 genes actually with a logfold change above 0. So we upload this Excel file in geneXplain platform. To upload any data, you have to click here, open it. It detects the format of the file and which column is ID for the genes. You then import this file. It's ready for your analysis. So to analyze these genes, to predict the transcription factor binding sites in the promoter of these genes, you have to select this gene or protein list and analyze regulatory regions, identify enrichment of transcription factor binding sites in promoters using TRANSFAC. Here you take your list of genes and it takes as a default the list of housekeeping genes as a control. And the promoters which will be analyzed are of the length 1 kb upstream from TSS and 100 nucleotides downstream. And now you just simply run the workflow. You can see the pipeline is running. So here below you see the progress of the pipeline, which steps of the pipeline are already over and which steps are currently running. Here is the result of the analysis, and here you see the table of predicted transcription factor motifs which are enriched in the set of genes. You see here the name of the motif, the logo, and the enrichment. Yes to no ratio, actually the ratio between frequencies of the motifs found in the promoters of these genes compared to the promoters of housekeeping genes. And here we see the p-value of that difference. So to have a better look, let's have a look at this table. So basically this is the same table which you can sort, for instance, by yes to no ratio, and you see that the top motifs found here enriched are the motifs for a real A factor, which are factors of NF-kappa B family. Actually, you see several of those motifs from NF-kappa B family are at the top of this enrichment. Another motifs which were enriched here for the transcription factors like SFOS and June, and for the PO factors. Let's have a look where are those sites actually located. We will now select the motif which is best here, and click on the visualization tool to see where those matches are found in the promoters of these genes which we are analyzing. All right, so you see here the list of these genes, and here you see the promoter areas. So the red bar is the first exon, and you see these red arrows are corresponding to the sites found in those promoters. So here we see them more in a better resolution. So for instance, in this promoter, the program found 2 binding sites for real A, and we see here the logo plot for this motif, and also we see the basically the core score and the score of that the full score of that binding to this site. Match is growing to something bigger, which is called match suite. We'll have a look at it, and the match suite looks for not just for enrichment on transcription factor binding sites, but actually pay attention to the tissue specificity of those transcription factors. And it helps to understand which transcription factors are actually active in the cells, in the cell types and tissue types you are analyzing. And it will open this match suite. In the match suite, we can input the set of data. For instance, in this case, we'll analyze a set of liver-specific genes. Here is the list of these genes. It includes actually 155 genes which were extracted from the data on Protein Atlas. So we will import this data into the system. For that, we will start the analysis. Select the human. We select the gene set analysis. And we upload the file. We then select this set of genes and click Next. And we run tissue expression based analysis. And we here select the tissue of our interest, which is liver. We should specify the region which we want to analyze, so we can extend these regions, for instance, to -1000 to +100, and then we click Next. The program is ready to start, so we launch the analysis. Results. You see the list of transcription factors which are found to be important for regulating of my genes, and at the top you see an HNF4 alpha, which is definitely very important for regulating liver-specific genes. And here we can switch to a more detailed view of that factor list. So it tells you which actually motif was used to find the binding sites for this factor. You see also this factor enrichment, which means the enrichment of these motifs in the promoters of that genes compared to the random promoters in genome. And you see the expression of that liver factor in the tissue of interest. Here you see the absolute expression value and also the relative expression of that factor in the tissue of interest compared to other 62 tissues in the database. So here at the right, you see actually the table of for all the genes which we analyzed, 155 genes. And here we see the table, which sites were found in the promoters of those genes, how many of those sites. For instance, in the first promoter of that gene, we found in total 20 sites, and there was one site for ATF4 and 2 sites for FOXO1 and so on. And here below we see actually the positions of those sites in the promoter of that gene. So here you see the gene, here we have the promoter area which was analyzed, and each of these arrows correspond to one found binding site in that promoter. Here at the left we see again the information about these sites, what is the score and what are the position of that site in genome, and what is the logo plot of that consensus of that site. We actually run the site enrichment using 2 algorithms. One is called just a site enrichment algorithm, which gives you enrichment of individual motifs in the promoters of those genes. And another algorithm is called composite module analyst, which is using genetic algorithms and pieces of artificial intelligence. And these look for a combination of transcription factor binding sites found in the promoters of those genes. And you see here that HNF1 sites are combined with P53 and also with HNF4 and other sites for other factors. TRANSFAC has also other information which can be called up for this gene. So here in this display tracks, you can select promoters and enhancers, also experimentally validated transcription factor binding sites, conservative regions in genome. So I click them all, and now you can see, for instance, here for this area of the promoter which was analyzed, we see here the experimentally proven sites which are collected in TRANSFAC. And for instance, here you see these FOX sites were identified here, and they're actually found to be near those predicted FOX sites by our analysis. We also see other transcription factor sites, like sites for glucocorticoid receptor or for CREP, which were not found here by our analysis, which actually specify basically the condition. So the analysis was done for the liver transcription factors, and in the we search for the enriched binding sites in the liver expressed genes. So the other binding sites which were identified experimentally in the promoter of that gene may function in different conditions. So that's why they were not found here by the site's enrichment algorithm. We see here also information about conserved regions in genome, and this gives you additional evidence that, for instance, this region is definitely very much conserved. And indeed, most of the sites which were found here and important for regulation of this set of genes are located in this conserved area. And also we can coincide the information with information about SNPs and variations in genome, and look which variations actually can be found in the sites, inside the sites, and may influence the sites, may change the consensus of those sites, and consequently may influence on the regulation of the gene. So we see here also enhancers, and to see them more clearly, we will zoom out, and we see here some of the enhancers, which are associated with this gene. And these enhancers are acting in neutrophils, in thymus, in blood cells, in brain, and we got this information from Phantom5 database. Actually, these enhancers can be also analyzed in a separate analysis. Another feature of TRANSFAC BASIC is a support of all major omics data types. Basically, you can analyze RNA Seq data, ATAC-seq data, ChIP-seq, cut and run. You can also analyze the whole genomic sequence or the partial exome sequencing data. You can also analyze single cell data in all of these omics data tab. If you have proteomics data, you also can analyze it using TRANSFAC BASIC tools. And the best part, you don't need to write any single line of code. Just upload your data, even the raw data, and pick up the right pipeline and simply go. Understanding combinatorics, how transcription factors work together, is a major challenge. Our machine learning-based tools help to uncover how these motifs co-occur, how they cluster together in the promoters of these genes, and how they do this synergistic regulation. And this is essential if you're working with data coming from complex diseases or with complex traits. Every tool in TRANSFAC BASIC is available through an intuitive graphical user interface. You can drag and drop analysis steps, adjust parameters, and get immediate results, all in your browser. Perfect for biologists who don't want to code, and powerful enough for bioinformaticians who, of course, do code. You'll also get access to over 200 bioinformatics tools and pipelines for FASTA and FASTQ file analysis, for VCF and BET files, for functional enrichment, for powerful statistical analysis, and even more. Everything is built to work together very seamlessly. So this is a TRANSFAC BASIC. It gives you the largest positional weight matrix collection available. Smart tools like match for accurate site prediction. Experimental evidences from ChIP-seq and other data, promoter and enhancer context, full omics support, machine learning tools, an easy interface, and the power to go deeper if you want to. If you are working in transcription regulation or trying to understand upstream control of the genes you are studying, this is a tool for you. You can learn more, just contact us at the geneXplain.com. Thank you for watching and see you soon.